I was talking to Sean earlier today.
Yeah.
He wanted me to run another incubator experiment, but instead of doing a polypropylene connection, I want to do a polypropylene double McMaster to silicone to asteroid. And then we're also going to be doing pushing fluid instead.
Okay.
So it's showed it was more stable.
Okay.
Only lives the trench super.
Huh?
The trench.
Oh, yeah.
One step closer.
Got trenches in the cartridge now.
Next we'll add walls in it.
Walls. We gotta build a wall.
How do we hide this owl? You want to hide what? This top bar.
More did you connect, no?
Hide floating meeting controls.
Hide
Next one. Yep.
I need new glasses, guys.
Yeah.
All right.
What's the frequency to get the ideas? Is it?
Oh, I
You
don't
should
know.
control like
It's
I
only
mean,
it's only when you feel like there's
Yeah,
high pain
it's,
or something?
when you go, I mean, me, they ask me to go every year, but
depends, I guess, when you're younger, since the that's decrease you know.
Okay, yeah.
it's more when you feel it.
Good. I mean, when you don't see, you have to check.
Yeah.
Okay, thank you guys very much for coming today. Welcome to our workshop on fundamentals of cell culture. Today we're covering aseptic technique. We have Lisa Resende, the Director of Teaching and Learning, who's going to be observing. And at any point, we encourage questions and discussion. So I'm going to, I've already shared my screen at the beginning. Okay, let's go. Okay, so to start, we're gonna take a formative assessment of what we went over last week. And the reason why we do this is it's proven practice of learning to recall previous knowledge and to gauge what you have learned and what you need to work on. So we're gonna take 5 minutes to work on the quiz that we have in front of us. And at 2:10, we're gonna come back and we're gonna grade the quiz together. This is a low stakes quiz. You're gonna grade your own quiz, and it's really just for you to know what you've learned from the previous lecture and what we need to go over again. I am gonna collect the quizzes at the end so I can also understand how effective that last lesson was. So go ahead and do your best.
Oh. Yo.
And if we finish early, just let me know. We can continue on. But feel free to take the full 5 minutes.
Mhm.
Oh. James brought in treats.
Oh, yeah.
Thank you.
Yeah,
Thank you.
have some treats.
I didn't get a peep this Easter, so thank you.
Yeah, you gotta try those peeps.
Just 10 chicks. Full of ducks.
Okay.
Interesting.
No, but
So good?
finished.
Okay. So let's go ahead and grade our quiz together. And if you have any questions or even discussion on the quiz itself, feel free to introduce your input. So question one, what do the numbers represent on the cell culture flasks called T25s, T75s, and T225s? The answer, the surface area of the cell's attachment inside of the culture flask. Do we all feel good about that, what those numbers represent? And we're going to go into that in our next session about why that's important, but it really gives you a good idea of how many cells you actually have in your flask, and it can help you to plan for future experiments when determining how many cells are required.
So the 25, 75 to 25, that's like millimeters,
Centimeters squared.
centimeters squared. Okay.
That's a good question. Thank you for that clarification, James. Cool. Okay.
So what does the T represent?
The T. Hi.
Is the flask shoulder. Well, there's many debates on what the T origin is. Some say it's the shape, but last call to Granger at least, or Corning, they mention it's because you have the angled nature of the flask. It's apparently something.
It looks like a T. So I don't know the answer to that question. That sounds very plausible though, Yvonne. I can definitely follow up with you later, James, because that will help us to construct new knowledge and build a map in our head of understanding. So thank you very much for that, James. Okay, so question two, this is a simple matching of the following. This is the different cell culture types as we presented them in the class. So tissue explants were the intact tissue collected from the organism, and then we grow it in the lab. The primary cells were the cells of the intact tissue that we dissociate into single cell suspension for culturing. The cell lines are established cells in the biobank that can be used in different labs for similar results. And then finally, stem cells are those master cells that replicate unlimitedly and differentiate into any type of cells. Do we all feel comfortable about that? Is there any questions about any of these cell types? There is definitely a lot of details in this, and your mentor will go into more depth on these different cell types and what will be used in your particular case. Yes.
Can tissue explants be preserved before they're used in culture after harvest, or do they need to be cultured immediately after harvest?
In order to have a really physiologically relevant, something that acts like the body, we need to use it immediately. So that is one limitation of tissue explants, is you have to go directly from biopsy, from the operating table or whatever kind of method is used, to the cell culture lab. And you have it's limited in hours. And you may be able to keep the tissue alive, but to have it metabolically represent the human body or the organism, it has to be quickly. So no. But your mentor will go into more depth with that as well. Okay, Shaivi just joined us, so we're going over a quick quiz from the last lecture. So just bear with us, and we'll start soon. Okay, so question 3: in addition to heat and humidity, a cell culture incubator supplies what to the sodium bicarbonate to maintain the sodium bicarbonate buffering system? The answer is 5% CO2 gas. Now, it's important to understand, though, not all cell types require this buffering system. Not all cell types require the sodium bicarbonate pH control in your cell culture media. This has actually happened to me before, where I used a certain cell line and I put it in my 5% CO2 gas incubator and it didn't grow and I didn't know why. And it turns out I didn't read the vendor's website close enough and it actually said, do not use 5% CO2, that the cell culture media is buffered without that. And if you use 5% CO2, you'll kill your cells. I learned that from one of our old lab managers, Julie, who had happened to use that cell line in a previous life. Are we all comfortable with the 5% CO2 though, and the sodium bicarbonate buffering system?
Yes.
Yes. Okay. And we have somebody who monitors that gas, so you don't have to worry about adjusting the gas. It's something that will be taken care of. Okay, and then the last one, name 2 visual indications of cell culture contamination, microbial contamination. So I listed several visual cues. The most common is turbidity, but this can also be shown through acidification, where the bacteria is giving off excess lactic acid and turning the cell culture yellow. You could have your cell culture line slow down. You could have excessive cell debris death, death of your cells, and you may see budding yeast or fungal like hyphae tree roots. Any questions on any of this? Okay, so I'm going to go ahead and collect these quizzes at the end of class, and that'll just help me to really understand where we're at. If you didn't get all of them, that's fine. It just helps me to change and adjust the class moving forward. Okay, so I'm going to do a prior knowledge check, and this is for me to gauge what the class already knows and what we don't know so that I don't make assumptions. And I'm going to also help you to activate prior knowledge to put you in a frame of mind for what we're about to talk about. So the question I'm going to put out to you, what is the difference between sanitization and sterilization? And if we don't know, that's okay. But yeah, anybody feel free to raise your hand and chime in.
It's a good question.
Yeah.
Sterilization, I would say, it's like you eradicate everything, maybe below a certain threshold.
Yeah.
And sanitization, it's more like you clean, you maybe remove what you have on your surface, or you inactive them without necessarily elimination.
Precisely.
Use serial dilution sort of an approach to reduce bioburden versus terminally sterilize below a certain CFU growth threshold. With sanitation, it can return back to the previous dirt condition.
Thank you. Yes. Shivy, in your life, in your common life, where would you hear the word sanitization or the root of that sanitize?
Well, I've most commonly heard it like when we're told to like sanitize our hands, like just to use like hand sanitizer.
Yeah.
Is that just like gets rid of like some of the bacteria on our hands, mostly like disinfecting. I guess sterilization would be like beyond that.
Do you hear that word sterile in any part of your life? Normal life?
In normal life? I don't think so. I don't think I hear it a lot.
Yeah.
In the lab, I hear it a lot, but
Yeah.
not outside.
Can anybody think of another place besides the lab where you might hear the word sterile or sterilization?
I think of was it sanitation workers,
Yeah.
people that do cleaning. I think of like sterilization of like maybe like in a hospital. I
Yeah.
want to keep like the room sterile or the equipment.
The equipment, especially the equipment. Yeah. If you're doing like brain surgery or something, that's highly important to keep it sterile.
Mhm.
So good. Okay, we covered it. We had Jerome give his definitions and Navon summarized them, and then we talked a little bit about where we hear it in our regular life.
Mhm.
Excellent. Okay, so our learning objectives for today, we want to identify the common sterile components in our laboratory, the consumables, the single-use consumables in cell culture. We should be able to accurately describe aspects of aseptic technique in cell culture practice. And then we should be able to identify poor aseptic technique in practice. And we're going to do proof of learning by doing knowledge checks, as we did before, and discussion throughout the lecture. Okay, so aseptic technique is basically the prevention of contamination within your cell culture. And contamination can be bacterial contamination, mold contamination, or it could even be contamination of other types of cells. Say you have a certain cell line, you could contaminate it with another cell line inadvertently, accidentally. And contamination could also be with unintended chemicals being added to your cell culture. There are several components that interplay with each other that collectively work to support good aseptic technique. I list them in this infographic circling aseptic technique, and we're going to spend the lecture going through each one of these components. So just take a moment to look at this. So the first one, sterilization. In cell culture work, we culture our cells with cell culture media. The cell culture media comes to the laboratory pre-sterilized. It's our job as lab workers to handle the cell culture media to keep it sterile. Occasionally, we have to add cell culture media additives such as serums, hormones, growth factors, and these may or may not be sterile when we receive them in the lab. This would be indicated on the packaging. If they are not sterile and we add it to our cell culture media, we have to find a way, we are required to re-sterilize the cell culture media. The most common method of sterilizing cell culture media is filter sterilization with a 0.22 micrometer pore-sized filter. This is where we run this liquid cell culture media through a filter under vacuum, which pulls the media through these pores and traps bacteria. There's debate, conversation in the field of cell culture that the standard 0.22 micrometer might not be small enough to capture mycoplasma. Mycoplasma, again, is that bacteria that's so small we can't see it even when we have contamination within our media. And it's the smallest bacterium that we know of, which is about 0.2 micrometers in diameter. So in order to feel more comfortable about capturing mycoplasma, some labs will use 0.1 micrometer pore-sized filters. In our lab, we're currently using 0.22, which is the standard. And this filter sterilization, again, is particularly important for growth factors, serum, and other heat-sensitive reagents that can't be boiled or cooked. So once we have our sterile cell culture media, we need to put it in our cell culture flask. Our cell culture flask that we perform our cell culture in comes to the lab sterile on the inside. And again, it's our job as lab workers to handle these flasks to keep them sterile on the inside. When we're moving large volumes of liquid in and out of these cell culture flasks, we use an automatic pipetter gun, which I show here on the left. This is basically a motorized vacuum that can either pull or push liquids into serological pipettes, which I show on the right. These serological pipettes, they're single-use, single-package, sterilized pipettes with markings of volume on the side. These come to us sterile. They have cotton plugs on the top that prevent the automatic pipetter from inadvertently adding contamination to our cell culture media. When we're handling smaller volumes of liquid, we use micropipettes. On the left, I show a pipettor, which controls the suction of liquid that is pulled up and pushed out of pipette tips, which I show on the right. These pipette tips may or may not come to us sterilized, pre-sterilized to the laboratory, but this is something that would be indicated on the packaging. Typically, though, in our lab, we buy pre-sterilized pipette tips. For cell culture work, we have to get rid of waste, liquid waste. We have to dispose of it. And the most common method to do this is with glass pasture pipettes, which I show on the left. This is a glass tube that we hook up to a vacuum line, and we pull our liquid waste up through the glass tube into a trap, which I show on the right under vacuum. These glass pasture pipettes are typically non-sterile, and we have to sterilize them in the laboratory. So how do we sterilize? When sterilizing a hard surface, such as glass or heat-resistant plastics, we use what's known as, we use a device called an autoclave, which uses steam, heat, and pressure to denaturize proteins, which kills living organisms. It's a very reliable method of sterilization. It's typically run at 121 degrees Celsius at 15 psi for 15 to 20 minutes. And we can do this for solids, like the glass pasture pipettes, or different tools like scissors and tweezers. And we can also do it with heat-resistant liquids, such as cell culture media or buffers. When we autoclave something, we typically have to have a vessel that we place our tools in. Here I show an autoclaved bag that seals under heat and pressure so that everything within the autoclave bag stays sterile after we take it out of the autoclave. We place autoclave tape on these vessels to help us determine what has and hasn't been autoclaved. This particular autoclave tape turns from white to black after being exposed to 121 degrees Celsius for more than 10 minutes. And that's a good way to keep track of what you have and haven't autoclaved. And this is particularly important when you have a lot of instruments in the lab and you have to keep track of what's sterile and what's not sterile. I've definitely done that in my career where I forget what is usable, what is sterile, and what is not. So I highly recommend using autoclave tape whenever possible if you have to use an autoclave. Okay, so for our first knowledge check, this is going to help you to recall what we just went over, and it'll help me to determine if we need to go back and review it. All cell culture consumables purchased from a vendor that come to us in the lab are pre-sterilized. So we're going to use a finger answer in front of your torso. Either put up a 1 for true or a 2 for false. Okay, we all said false. That's correct. So as I mentioned, there's different additives to the cell culture media that may come non-sterilized, or pasture pipettes, or even the micro pipettes that we use. Sometimes we buy non-sterilized ones to save money. So very good. Okay, so I'm going to encourage a little more discussion here. This is called a think, pair, and share. So you're going to pair up in twos and share your ideas on what we have going on. So based on your current knowledge, would an autoclave be a sufficient sterilization method for all cell culture needs within our lab? We're going to take 5 minutes to think, pair, and share. So at 2:30, we're going to come back and continue on with the lecture. And I want you to try and recall what the different consumables are in the lab and what the requirements are for sterilization. And then even taking a step further, could you imagine different scenarios in the lab where we may have to re-sterilize things? So let's take some time to think about it.
What do you think?
Okay.
I'm speaking English, sorry.
All cell culture needs. I would not say all cell culture needs, but probably.
Yeah, good intuition. I think so.
Maybe I said no.
Why?
What you're using
For tradition, I can definitely sterilize cells, but I think stuff like.
That's the thing.
Worms,
I don't know.
I don't think it's sterilized.
No,
For.
But the only kind of sterilization we.
Yeah, I think.
That's why there's other methods like,
Material like
Irrigation.
radiation.
A
You
sterilization.
have that method.
Belt,
Well, with the radiation, it doesn't
nature, you know.
require.
You're right, that you think we use.
Yeah.
We have to.
But I think it does get
Some of them.
levels hot.
And
So I think if you just do plastic,
if we really need to.
remains at room temperature.
We need to find another way to sterilize.
That all the.
The autoclave, it's nice, but the goes down, you know, but it doesn't work for.
Autoclave media.
Typically, when we sharing, usually we use also, you
I don't know
know.
if you would do that in addition to.
For the
Standard filtration.
They are already.
Mhm.
Or if it.
They are well, but
Yeah.
if you need around.
Mhm.
I don't know because it's.
So utilizing media.
Yeah.
That's not a
sterilization. Paper projects are like.
That's using.
So sometimes we have additives
So,
that we
yeah,
put
I
in
think
cell culture
that-
media
Right.
that will disassociate
I
the
think
nature
noise.
at high temperature of the autoclave. So there's in autoclaves certain additives like serum.
Compatible for everything.
Your blood,
Here we are.
you put that in the autoclave, it all just
The face,
ripped
but
apart and no longer useful for the cells.
What
So
about,
that
you
is,
know,
that's
like,
when we use the film.
or even the medium that.
And maybe some of the
Can you really
devices
put
we're
that in
using,
the autoclave?
they'll probably get
Can you
damaged in the
really
autoclave.
denature all the protein also. you
But that
have
barely
medium,
happens.
You know, and then.
But
Have you seen it happen?
no, but I guess when we're prototyping,
Yeah.
yeah, it's mainly when our prototyping. So we do stuff with the 3D printed material. It gets damaged.
What for the
Yeah,
medium?
I've definitely seen it.
Yeah, in the jar,
Okay.
yeah, that would be.
And then
I mean, that
we said
would be
earlier too
really
that
glass bottle.
there's some
But then,
contaminants
you know,
that don't
might.
go away after autoclaving. I think you mentioned spores one time.
Yeah.
Spores survive.
You ever,
So autoclaving
autoclave
is the
medium?
preferred method for taking care of spores.
Medium,
It's one of the
yes.
few methods.
Yeah.
After.
Ethanol.
No,
Or
yeah.
hydrogen
Yeah, I think I
peroxide
tried
will
once
not
with the
majority of
film, it was all.
Is,
Okay.
yeah.
Oh,
sterilization.
it was, yeah.
Teenage is all.
Yeah,
zero.
the nature, like.
I've done
Gamma
even
irradiation.
alone,
Ruthenium,
I mean,
ethylene oxide.
yeah, without serum,
Sterilization.
but with.
Can a tardigrade survive
example,
autoclave?
LB
Well,
brought,
actually, I'm not.
in my
I don't
past
think so.
life,
Tardigrade,
yes,
no.
when I used
They're
to
very
do
good at
that,
surviving.
LB
radiation,
brought,
but not.
Maconi agar, all of those can be, they have pectin in them.
There's a lot
That
of board
typically
that's lived in the
survives,
dirt.
but most of the.
Hmm.
Or, I'm sorry, that.
Yeah, so if your cells ever get contaminated, just put them in a glass
getting mixed.
beaker and autoclave them, you can sterilize, clean yourself.
Right, that's
They just won't grow after, so that's the only drawback.
Yeah.
Okay, guys, I think that was, that's our 5 minutes. Let's go ahead and come on back. Did we have any interesting discussions that we want to add to the group? Anything that you didn't expect to come out of that? It's okay if not. Yes, Navan.
We were wondering if our cells get contaminated, we could put it in a glass beaker and autoclave the cells to clean them and then reculture.
So the way the autoclave works is the heat and the pressure cause the proteins in the cell to denature, to split apart, and everything just kind of rips apart in the cell. So you're going to kill everything in those cells,
Oh, no.
in that cell pellet.
Can you cook food in an autoclave and eat it?
Yeah, it's a lot like a pressure cooker. My grandma used to have this thing that it was like a vessel that you'd put in the microwave with water and food, and it would pressurize it and it would cook it much faster. So yeah, you could definitely cook in an autoclave.
And it'll sterilize the food.
Yeah.
So they do
Might not.
pickles,
Pickles.
pickle jars with pickle in it, they put it into a, what do you call it?
Pressure cooker.
Pressure cooker and
Yeah.
terminally sterilize it.
Cool.
Yes.
Interesting. Okay. So moving on to our next component of aseptic technique. We have environmental controls that greatly reduce the chances of contaminating our cell culture. Here on the left, I show a picture of a dirty laboratory. This is very exaggerated, but at all times we have bacteria on our surfaces. And depending on how we treat our areas, it will have more or less bacteria. So there's always going to be accidents in cell culture. But even if you have an accident, that doesn't necessarily mean that your cell culture is going to become contaminated. So the cleaner you keep your area, the more organized and the more the faster you clean up messes, the less chances you'll have of contaminating your cell culture. We always want to clean before and after our work, and we want to keep our area clean while we work. In addition to cleaning your area, it's important to wash your hands and remove jewelry before you start working on cell culture. This reduces the amount of bioburden that you bring into the laboratory from the outside environment that you can't control cleaning as well. We don't know how often people clean their hands before touching doorknobs. We don't know what's going on different keyboards and computers. So we always want to wash our hands and remove jewelry before working in cell culture. Yes.
Just quickly, I believe you're gonna talk about it, but you do wear gloves probably for the cell culture, right?
Yes.
You still need to wash your hands even if you do have gloves.
It's best practice to wash your hands before you wear gloves because you're going to be touching other things in the laboratory without a gloved hand. So, you're going to open and close the door that you may touch a glove with as well. Or you may touch other things in the laboratory like handles or equipment. So it's always good to wash your hands. It's just good lab practice.
Or your glove rips.
Yeah. But yes, that is a good point. So gloves are not enough. Always want to wash your hands. And we'll talk more about washing hands later in this lecture. The big one that I see most people make the mistake of is leaving their jewelry on. I've worked with people who refuse to take off wedding rings. And if you come to that point, it's just something that we'll have to work around. I understand if you just absolutely won't take off a wedding ring or whatever, but you should take off rings, watches, wristbands. Okay, so the majority of our cell culture work is done within a biosafety cabinet that we commonly refer to as a BSC. This is an environmental control. A biosafety cabinet is basically a cabinet with filtered air in it. We have a glass sash that comes over the opening with just enough space open for us to place our arms in and manipulate the cell culture. A biosafety cabinet basically consists of a blower motor at the top of the cabinet that pulls air into it and pushes this air through a fine particulate filter known as a HEPA filter. This filtered air is pushed down towards your working space where you'll be doing your cell culture. The filtered air is pushed either to the front or the back of the biosafety cabinet and pushed down into a grate where the air is recirculated back up to the blower motor and back into the HEPA filter to be refiltered. Some biosafety cabinets will exhaust a certain amount of this air. But this is how we keep our air clean when working with cell culture. Like right now in this room, it's hard to see, but there is quite a bit of dust. And on that dust, we have bacteria and mold. So this is how we try to minimize any of that dust or bacteria particulates getting into our cell culture. So I'm going to go ahead and show a picture of a smoke test in a biosafety cabinet, and this will give you a good idea of how this works. So here we have a smoke apparatus inside of the biosafety cabinet and a smoke detector on the outside, and they're showing that no smoke is leaving outside the biosafety cabinet. It's all being pushed into the front grate right now by the air being pushed down from the filter. So when we take the smoke device outside of the biosafety cabinet, we're showing that it's all going into the grate and none of it's going into the working area of the biosafety cabinet. And that's the front grate. There's also a grate on the back side of the cabinet. If you can see in the back, those little holes, that's where half of the air goes to the back and the other half goes to the front. Cool. Right. Now he's showing, this person is showing putting their arms into the biosafety cabinet, still has the smoke going into the grate. It doesn't affect your working area. It's pretty good at pushing all of the air. So when you go in and out of the biosafety cabinet very fast though, this can disrupt the airflow and potentially guide some of these particulates into your working surface area. If you cover the grate, that's a different story. It disrupts the working surface area and you can have quite a bit of disruption of filtration occur. So we have particulates from inside the biosafety cabinet escaping, and we also have particulates from outside getting into the biosafety cabinet. So it's really important that we keep that front grate area clear of any debris. Yes, the bottom.
So you cannot work in the front grate area, correct?
We should avoid working in the front grate area, yes, and especially avoid covering it. If your arms touch it a little bit, it's okay, but really best practice is even to elevate your arms. And the big one is not to go in and out too fast, especially when you're in a hurry. We get that temptation.
And you can put things back to the, no?
That's a very good connection. Yes, we should not cover that back grate. That also disrupts the airflow and increases your chances of contamination events. So some basic rules of aseptic technique. Always keep your containers closed whenever possible, so that if you do have contaminants that get into the biosafety cabinet, at least it's not getting into your sterile surface areas.
Question.
Yes, sir.
So this arrangement, is this okay because it looks like those pipette tips are blocking the back area? Would that disrupt airflow or does it matter?
No, it does matter. Honestly, I did not see those pipette tips. You're talking about the ones off to the right-hand side by
Yeah.
the microscope?
Mhm.
I didn't even see that. Yeah, you're right. That is blocking. No, that's not an appropriate arrangement. Good catch. I would even say that microscope is kind of iffy about whether it should be in there.
Where should it be at then?
There should only be one box of pipette tips at a time. You should minimize the number of pipette tips you have in there, and they should be in the center area, off to the right or left. Very good catch, James. So when working in the biosafety cabinet, you should only work with one cell line at a time so that you don't inadvertently cross-contaminate your cell lines, and you should stay organized. This is a very cluttered biosafety cabinet, and the chances of making mistakes exponentially increase when you're working with so many different components. And a lot of it is probably not sanitized as well. I see paper in there. I see a lot going
Yeah, I
on.
mean, do it directly on the bench top,
Yeah,
It's useless.
yeah, this person's doing some strange stuff. And it also speaks to other people about the quality of your work. When somebody comes in and they see your work surface looking like that, they also will question the type of work that you're doing, the quality of it. You always want to avoid crossing your arms over open containers. So if you have a contaminant on your lab coat and you have air blowing down, it's possible that that air will cause the contaminant to come off of your lab coat. If you're over an open container that you want to keep sterile, that will be a contamination event. The best way to avoid this is to not cross over open containers whenever possible. And then lastly, it's important to always clean up after yourself as quickly as possible. If you have a small media spill or a small spill, you can use 70% ethanol. You just spray a wipe with 70% ethanol and wipe it up immediately. If it's a major contamination event, we use 10% bleach for 10 minutes. I have a little infographic of how you would do this. You would douse a rag with your 10% bleach, place that rag over the contaminated area, let it sit soaked for 10 minutes, wipe it up, and then dispose of it properly. And then let your supervisor know what's happened. Let your mentor or supervisor know what's happened. Our last barrier of preventing contamination in your cell culture is antibiotics. The most common antibiotic used in cell culture is penicillin streptomycin, which is effective against most bacteria. But there are several types of bacteria that have resistance to penicillin and streptomycin, such as fecal matter bacteria or mycoplasma. If you need better, stronger antibiotic than penicillin or streptomycin, another option is gentamicin, which is a broad spectrum antibiotic. We want to minimize the amount of time that we use gentamicin because it is more cytotoxic to your cells. It changes the way that they behave.
Mhm.
And then if you have a concern of yeast or fungus, we can use amphotericin, which also is rather cytotoxic to your cells. And we want to minimize the usage of this whenever possible. Many labs will not use antibiotics at all because it does change the way your cells function. But it is normal practice for laboratories to incorporate antibiotics in their work.
Wait, you mean like my cells in my body or the cells?
The cells that you're working with
Okay.
in cell culture. You can add antibiotics. It's an additive to your cell culture media, and it changes the way those cells behave inside of the flask.
Oh,
And
okay.
And it depends on the type of experiment you're doing. It may not matter if you have antibiotics, but if you're looking at different signaling pathways in your cells that could be disrupted by antibiotics, that's something you want to consider. And your mentor will have direction for you on this. They'll be able to tell you if you use antibiotics and if it's appropriate or not. So do we have any questions about that, about our lecture thus far?
One question.
Yes, sir.
So the hoods, they come with a UV bulb?
Yes, so they have
How
a
do you use that?
so it's UV irradiation, which breaks apart DNA, double-stranded DNA breaks, which hypothetically will kill organisms. It's been under the recommendation of NIH to not use UV for sterilization, because UV does not pass very well through different surfaces. Like if you have a plastic tip box, the surface area underneath the tip box will not be exposed to the UV radiation. Or if anything comes in the pathway of the UV radiation, you won't have sterilization. So I didn't talk about UV radiation. It's something that NIH is not recommending anymore. We should not rely on UV radiation. And then we also have to have a maintenance schedule for the UV bulbs to double check their quality and effectiveness. And a lot of labs are not equipped for that.
Cool.
Cool. So I'm just gonna leave a little bit of silence for us to think if we have any more questions. Sometimes the best questions come in silence and uncomfortableness.
I mean, the follow-up question to James would be then, how can you clean your biosafety cabinet then?
That's a great question, and that's a good lead in to our next activity.
Good.
We're gonna go ahead and show a video, a good aseptic technique. And then we're gonna follow up with a video of bad aseptic technique. And then I'm gonna ask you to identify the factors of the bad aseptic technique. And that'll be a way for me to assess how good the learning was in this lecture. So I'm gonna go ahead and narrate what's going on here. When entering into the cell culture laboratory, we start with washing our hands and removing jewelry, such as a watch or rings or bracelets. So washing your hands isn't just merely getting them wet and adding soap. It's rather methodical. It starts with first wetting your hands thoroughly. The water really needs to get in all of the crevices of your hand, your fingerprints, your handprints. And then you need to foam your hands methodically, getting all surfaces. That actual physical friction motion is what's used to break apart the oils and different bacteria and contaminants, organic contaminants on your hands. So he's washing the backs of his hands, the palms, his fingertips, his thumbs, And this is a rather long process. A lot of us can get in the habit of washing our hands way too fast and not really being effective.
And what about under the fingernails?
Under the fingernails, it's that practice where we balled up our fingers and then pressed it against our palms.
Hmm.
I'm demonstrating it right now.
Hmm.
We want to consider trimming our fingernails as well, especially when working in cell culture. We don't want to have our fingernails too long. So we'll dry our hands to not spread soap or water across the lab. This is followed by putting on our lab coat, and we see that we pull the cuff of our lab coat over our palms and then bring our lab gloves over the cuff so that we don't expose the skin of our arms to the working surface area. So this is intentionally set up this way so that the clean lab coat is being exposed to our working area. So we open our biological safety cabinet and we wait 5 minutes for the airflow to stabilize, because when we come to it initially, it's not flowing at its full rate. We want to wait that full 5 minutes. So we're going to pretend that in this video, we waited the full 5 minutes. We'll start with cleaning up our working space by moving all the equipment to one side. And then we're going to spray down our working surface area with 70% ethanol. Now this is methodical. We don't just spray down and do a quick wipe down. We're actually going to spray and wipe down the entire surface area. I'm going to speed up the video here, but I just want to show you how thorough this step is. A lot of people can fall into the practice of skipping this critical step, but you really want to clean everywhere. And he's generously wetting his wipe and methodically covering all of the surface with this wipe. And the reason why we use a wipe and we don't just spray, the ethanol, like soap, requires the frictional force to tear apart proteins within the cell membrane of bacteria or fungus. And some bacteria are actually resistant to 70% ethanol. So the ethanol has the polarity where it will, like a magnet, it will pull the bacteria off the surface with the friction of the wipe. So with bacteria that are resistant to ethanol, you physically need to wipe down the area to better sanitize. The ethanol is not a sterilization method, so there is still bacteria in there, but you have greatly cleaned it. And again, this will decrease the risk of inadvertent accidental contamination. Because accidents do occur. Everybody makes mistakes in cell culture where they accidentally touch their sterile tip on a dirty surface or on a non-sterile surface. But that doesn't necessarily mean that you've contaminated your culture. It just means that you need to follow up with your supervisor and do surveillance for bacterial and other microbial contaminants. So he's methodically going through every piece of equipment and wiping it down. Wiping underneath surfaces, wiping down all pipettes. And this is sped up, but this process alone should be something that you should have time management for. You should set aside at least 5 to 10 minutes to clean your working surface before you start your work. If you don't have time to clean, you probably shouldn't do that work that day.
You do that before and after.
Yes, sir, before and after. And you should always treat your working surface area like the person before you did not clean, because things happen. Things do happen. That's the vacuum line that we use for aspirating cell culture media. He's cleaning the vacuum line knob and the hosing. And then lastly, the grate area where we place our arms. We're rearranging the working surface area to go from dirty to clean, or I'm sorry, from clean to dirty. So Naban, or the person in this video, is right-handed. He's placing all clean material on the right-hand side and then going from right to left. where dirty material goes to the left. Anything that goes into the biological safety cabinet is sprayed down with ethanol and wiped down before it goes into the biosafety cabinet. This is not sterilization, but it is cleaning it to reduce bioburden for accidental inadvertent contamination. That's our serological pipette that we went over earlier. That's going to be sprayed down. It's individually wrapped. It's sterile on the inside of the packaging, but on the outside, he cleaned it just to reduce bioburden. So when opening containers, we only want to open one container at a time. We want to minimize the open containers. So if it doesn't have to stay open, we keep it closed for as long as possible. He's avoiding reaching over or stretching over open containers or stretching over at all. So he's passing items from one hand to the other. And you see how he loosened the cap but placed it back on the open container. That's so that if any unintentional contaminant comes into the biosafety cabinet, at least it won't go into your sterile working area. So he's opening his sterile serological pipette and he's going to take it out in a manner that where it won't touch any non-sterile surface. The cell culture media inside of that tube right there is sterile, so it's only touching sterile areas. You just transferred sterile media from one sterile container to another sterile container without touching any non-sterile areas. And then lastly, how we work with the cell culture flask. Again, we have our tubes closed whenever possible. He's taking a sterile pipette out of a sterile packaging and not letting it touch any non-sterile areas. He opens the tube and then closes it immediately. And then his flask is closed. He's going to open that. After he closed his media bottle, he's going to add his cell culture media. Perfect.
Is that a T25?
Yes, that is a T25 flask. Very good observation. And notice how he's also working in the center of the hood. He's not working on the edge close to where he could potentially have particulates come in from outside of the biosafety cabinet. Yes, sir.
Why did he put it in that separate conical vial instead of just putting it directly in the?
It was warmed.
That's a good question. It was just merely to show how you transfer liquids from one tube to another. There was no experimental reason. We just wanted to demonstrate what that would look like.
Makes sense.
Yes, sir.
Quickly want to add, one reason you would do that would be if you were wanting to heat the media. A lot of medias have extremely sensitive growth factors that typically you do not want to heat for a long period of time. One way to minimize the number of heat and chill cycles that a media undergoes, especially something like L-glutamine that can degrade into much more toxic compounds. The media would be you take a aliquot for that particular use session, heat it, use it, keep the rest of it in the person.
Makes sense.
Okay, and then lastly, disposal of contaminated materials. You're going to pull everything out that was used in your cell culture, place it in a biohazard trash, and then wipe down your area after working. So the same method. I'm going to go ahead and speed up through this just for time's sake. So here we're going to show bad cell culture work. Here, I want you to raise your hand and we're going to talk about every bad instance that you see, okay? And this is my way of determining if we can appropriately identify for aseptic technique. So what do you see?
The watch is on.
The watch is on. Why is that important?
You want to take off jewelry.
Yeah. Why do we take off jewelry?
Minimize bringing anything in.
Precisely.
It seems, I mean, we don't know, maybe the person wash his hands, but maybe not.
No, the person definitely did not wash their hands.
Touch your hair with your glove.
Did he just touch his hair with his gloves? What's in hair? Mycoplasma. Yeah, that's the most common area that you will have mycoplasma from.
Is it an issue of your body?
Unless his hair sterilized.
You put the white coat,
You put ethanol in his
the
hair.
white coat on
Without the,
after the gloves.
Yeah.
And the gloves are not over the white coat.
Exactly. Yeah, we can see that the
Skin
white
is
coat
exposed, yeah.
is not covering. So maybe that lab coat's a little too small. You should consider getting something a little longer and pulling it up over his palms so that the gloves cover his skin.
Oh, you didn't let the hood.
It didn't.
You guys are hitting him. Yep. So we didn't let the hood come to stabilization for 5 minutes.
I needed clean the tips before entering the.
Saw he just pulled those tips right into the biosafety cabinet. Remember, we were spraying those to sanitize them, to knock down the bioburden.
bench was not clean.
And he didn't clean.
Bench was not clean. This guy is really in a hurry. He is going too fast.
Media just sitting on the bench. I guess.
Media is just sitting in the bench. Yeah. Organization. Whoa.
Didn't sanitize
Everything.
the outside.
Yeah, the serological pipette was not sanitized.
Did touch the, yeah.
Did he touch the tip? He did. That's a sterile pipette, a serological pipette that is no longer sterile now.
Two containers open.
2 containers open. You are very sharp, Alex. Yes. Multiple containers open at the same time.
And you're like hovering over an open container too?
Hovering over the open, yeah, so, and why is that important, hovering over open containers?
You don't want debris from your glove or jacket to fall into an open container.
Yeah, and because it's in a biosafety cabinet, the air is flowing downward, that's a common contamination event.
Oh, they are a spill.
Spills, what's wrong with spills?
Well, they can create another source of contamination. They should be cleaned immediately.
Immediately, yes, you stop your work and you clean that immediately.
They left the tips open, no?
The tips are left open, so if you're accidentally spraying stuff, if there's aerosolization, you're getting your tips contaminated as well.
The non-sterile pipette is going directly into the media part.
Uh-huh.
Should the pipetter be put on the rack after use or? It's
Yeah.
not okay to.
That would be better, I think, yeah.
I agree. That is a very disorganized working space. There's dirty pipettes, there's a pipetter. It's all just kind of flung around. There's not much methodology there.
Working in the grate.
Working on the grade area. Oh, he's almost not even in the biosafety cabinet now. Remember that smoke video of what happens when you cover up the grate or when you work too close?
Yeah, that's the.
Picking up a serological from the desk.
Yes. Yes, so.
Oh, yeah, you
serological-
reuse the dirty air.
It was dirty. It was
Yeah.
touching the work surface area. So this, it wasn't cleaned with ethanol. The work surface area before. Ooh,
Well,
Yeah.
when you open those smaller pipette tips, I'm assuming you said it comes from the factory sterile,
yes, sir.
but once you open it, it's not sterile. Could you autoclave it after opening it? Just sterilize
Yes.
it again?
So that type of plastic, it's polypropylene. It is heat resistant. You can re-autoclave them. but we tried to avoid re-autoclaving. We want to use best aseptic technique as possible, only opening them in the biosafety cabinet.
The one we use tip with the filter, right? It's autoclavable.
The filter tips are autoclavable, but I have experienced where the filters pop out after autoclaving.
Yeah.
So that's why we want to just avoid that as much as possible.
And some of them will actually melt and the
But-
pore size in the filter decreases, so then when you prepare viscous stuff, it's not accurate.
We ran a little bit over time, but this concludes session 2 of cell culture. I greatly appreciate you guys very, very much. Thank you so much for coming. Do we have any questions or closing comments? I'm going to come around and collect your quizzes, and that's just to help me gauge how well my teaching was in session 1. And we'll have a similar activity in session 3. We have one more session that will wrap up cell culture. So thank you all.
Thank you.
Sean.
I really appreciate you all again. Thank you.
Thank you, Professor Dunn.
Instructor, colleague.
Thank you, Sensei.
No. They talk a lot about learner-centered teaching as the instructor being a coach. It's not about what I do, it's about what you guys do and how well I can help you get there. So I know I still have a ways to go, and it's much harder in science education because it's very fact-based.
Mhm. Thank you.
Yeah, thank you guys. I really appreciate you all. I'm getting there.
Nice. They're really popular. I'll put these by the microwave. You can put it in your coffee too.
No,
Marshmallows in the coffee?
no, it was a hot chocolate.
Do you have feedback now? I got feedback from the first session. Yes, first one. Oh, I'm getting a teaching certificate, and this is like the last thing that I have to do.
Very good though, very good class. So much good. Very good. Oh, very good.
Oh, very neat.